Okay. Okay, it's almost 4 o'clock. So, hi, sai, konnichiwa, hello, welcome to the final session of the day and the final sort of talk sessions of the conference. So today we're going to be talking about bioblitzes and museums and how they are natural partners. Right. So I'm starting off the talks, and then we've got 3 online pre-recorded and 2 more live presenters for you. And then at the end of the session, I'll give a little summary of how the actual BioBlitz itself that we're running is going. Okay, so as I said, I didn't say, my name is Mike Rutherford. I'm Curator of Zoology and Anatomy at the Hunterian at the University of Glasgow. But I've only been in that position for 3 years. And before I was there, I was at the University of the West Indies in Trinidad and Tobago. And I was in charge of the Zoology Museum for there. And after I'd been in Trinidad for a couple of years, I finally got around to holding my first BioBlitz there in 2012. It was the first BioBlitz for the country, and as far as I'm aware, it was the first Caribbean BioBlitz. I would love to know if anyone knows of any earlier dates, but so far that's what I keep claiming. And it became an annual event, which was very nice to see. It was proved to be very popular. It was generally organized by myself and the members of the Trinidad and Tobago Field Naturalists Club, the TTFNC, and other staff members in the University of the West Indies Department of Life Sciences, but also many other partner organizations over the years. So just to give you the basics of what we did, in the first few years, we'd find a base camp. I'd normally just choose a particularly interesting site. We'd have a 5-kilometer radius circle for surveying around the base camp. I'd then start organizing teams and get those experts to organize their small subgroups, one for herpetology, for mammals, one for birds, one for plants, one for fungus, and so on. And then it would be a case of promoting the event using various methods I'll go into later. And then we'd head out to the weekend. The timing varied quite a bit, sort of September, October, November. And we'd start surveying normally about lunchtime on a Saturday, and then go for 24 hours. And it'd just be normally the experts, the surveyors themselves present on the Saturday. And on a Sunday, we invite the public along to join in, to see demonstrations, talks, and so on. And then finish up surveying at lunchtime on Sunday and make an announcement of final results on mid-Sunday afternoon. And you can see the red circles show the various locations that we went over the years that I was running the event. So we really went the full length of the country from Icacos at the bottom of Trinidad to up to Charlottesville in the top of Tobago. I'll just give you a quick summary. The first event was in Tucker Valley. We found 628 species, a nice mix of a little bit of coral reef and various forest types. Next event was Arima Valley. Some of you may know it's the home to the Acerite Nature Center, and that's where we were based, 771 species. Nariva Swamp, which is a wonderful site, mangroves and various other habitats out on the east coast of Trinidad, 742 species. And then our biggest one was Charlottesville up in Tobago, over 1,044 species, and that was part and due to the very good coral reefs all nearby. And then Port of Spain, we did a BioBlitz in the capital itself, which surprisingly has a lot of stuff very close by, 762 species, and we even had the president of the country drop by and say hello. That's him standing in the back in the middle with my daughter in the front. And then Icacos down in the far southwest of the country, 750 species. And all those events, we had many different partners. I think this covers most of them, but who they were, it depended on where the event was based as to who would be most relevant to have on board. But we had many repeat groups that took part, things like Trinibats, a bat-focused group, obviously. Serpentarium were a wonderful group of folks who did lots of herpetology-related outreach throughout the country. And then we had long-established groups like the Acerite Nature Center, who provided a base camp, government-related groups, Ministry of Agriculture to help provide for permits and access to certain sites, the Environmental Management Authority and the Institute of Marine Affairs both took part. And then older conservation groups like the Pointe-à-Pierre Wildfowl Trust, and El Socorro Center for Wildlife Conservation, and then newly formed groups like the Eco Divers and what was the other one? The ERIC, which is a wonderful organization based in Charlottesville. So lots of different groups took part, and the way they contributed varied from being base camp hosts to providing handling experiences, granting access to land, giving permits. And the organizations were usually approached by me just saying, do you want to be a part of this? Do you want to help us? And generally everyone was very keen to get involved. Sponsorship normally came from a few groups, and we got very lucky early on that I made a good relationship with a local bank, First Citizens, who actually had an environment outreach community program, and so they were already set up to provide funds, and they very quickly sort of realized that this was a very nice way to promote their brand, and I was quite happy to take their money. We also had some groups like Bermuda's Biscuits who would give up supplies of snacks and things. I always found it very useful reaching out to small food suppliers going, can you help our starving surveyors? So, and we also had a good partnership with the Green Screen, which is a local film festival, and we got to promote some of the films that have been made by participants over the years. Next, what happened was I left in about 2018, and fortunately I was very glad to see that other people took the event and kept it running. And the first time was in Toco, and they got 906 species, so a nice event there up in the northeast corner of Trinidad, and then into the heart of Trinidad, Tabaquite, in 2019, 674 species. And this was fantastic for me, being back in Scotland at this point, seeing the whole event happening without me was just, yes, it was a nice legacy to have left behind, but glad it didn't need me for it to happen. But then in 2020, you all know what happened, and it was a worry what's going to happen to the BioBlitz, but they just moved online altogether. They did a fantastic job. Thanks to iNaturalist, they could just promote it as a backyard edition. And so they did a great job of getting word out there to people through social media. And over one weekend, they had over 6,000 observations, 1,300 species plus, and over 214 observers. There were quite a few planted plants and captive animals and cats and dogs. It wasn't obviously all wild stuff, but it was still the kind of generating the interest. They carried that on again in 2021. By this stage, the country had opened up a little bit, but you still couldn't have big gatherings. So they decided just to carry on with doing it online. But people could travel to some of the previous places where BioBlitz has happened. And they had over 2,900 observations over 1,000 species. And then carried on online the following year. But this time they opened it up to other partners in the Caribbean. And so they brought in Jamaica and Barbados. Main reason for it was because the University of the West Indies, who by this point really the main people running it, is a multi-island campus. They have campuses on Barbados and Jamaica, and so it's like inviting in our fellow university people. Over 4,700 observations, over 1,300 species, and 124 observers. So it was nice to get a multi-regional event going. But then finally they got to go back to base camp. In 2023, they made a second trip to Tobago, up to Bucco Reef, which is a very famous area down the bottom of the island. Lots of wonderful coral reefs there, 899 species. That must have been so frustrating to not have just that one more species you could have added on. And then Guayguari, which is down in the bottom corner, nestled amongst lovely forest and lots of oil formation and oil pumps. So it's a fascinating site down there, 768 species. So that was just a quick look at what the events were, and looking at some of the technology that we've used and how it's changed over the years. iNaturalist didn't really have much of a following in 2012. I'd only just started using myself. I think I added about 7 observations to the first event, but I don't think anyone else used it. But then as you can see over the years, it's changed hugely in how it was used, to over those 6,000 observations for the backyard edition. And we do try and encourage most participants to share their observations on iNaturalist, but you still have people who prefer, here's the list of birds I saw, or here's my plant list. And so they still took the observations in different ways. But from my point of view, it's fantastic because I can still participate every year by being an identifier. And obviously it opens up to lots of other international identifiers as well. And you can see there 630 identifiers contributed to the event when there's only 3 times the amount of observers. For the museum side of things, we quite often collect invertebrate specimens in particular for further identification, and I'd make a few travel displays from existing specimens, and these proved very useful throughout the rest of the year. And this was another part of the project that proved useful. Every event, I'd create a big 6-foot by 3-foot banner that could be used at events out with the BioBlitz. And this is the way we normally set them up. And I did a little quiz to go along with it. And it was a nice way of getting people to look and see what else had been found in previous events. And they carried that tradition going. So we've got almost more than a decade worth of banners. I'd make things like spotting guides to be handed out at the events, and trails, normally a simple word trail aimed at quite young kids, but to be helped by their older siblings or parents. And these were used again during the year, for the rest of the year, by the Zoology Museum. Activities at Base Camp include informative displays by the museum itself, but by the Field Naturalist Club. Lots of other members, lots of our participants came up with all sorts of wonderful displays of fruits of the forest, mushrooms, snake skins, all sorts of things. Experts were on hand to discuss all sorts of identification with the public. Here's one of the marine biology group showing some of the fish that they temporarily collected. Arts and crafts are a popular part as well, especially colouring in is always a nice thing to keep the kids entertained and something they can go home with. Animal encounters, we had the zoo involved a couple of years. We had one of the rescue organisations involved, and they bring along animals that people could see up close. But one of the most popular things was definitely microscopes. People love looking down microscopes if you've never looked down a microscope before. Kids will love them. We'd get specimens that we were finding out in the field, put them on there. So the experts would use them first on the Saturday, and then we'd let the public get access to them. Big advantage of having a university full of old microscopes that you can use in these events. And the publicity, obviously social media, one of our main avenues, Facebook and Instagram, Twitter, all used to promote it. We've got a dedicated Facebook page for the event that's just kept on being reused over the years. We get on television quite often, normally get on the morning breakfast show in Trinidad and promote the event during the week. Old-fashioned print media was very popular as well. To begin with, we'd provide print copy for the media, but later on, quite a few of the newspapers might send a journalist to cover the event, which was nice. And then the Field Naturalist Club dedicated its final quarterly bulletin of each year to summarize the event. And the various heads of the teams would write appropriate summaries of what had been found. And all these things pulled together and catalogued and kept together on a dedicated website as part of the University of the West Indies. And so all the links to everything that's produced can be accessed in one place, including a copy of the banner. For example, a school wanted to have a copy for their own use. And then finally, a final report, all written by me to begin with, and then passed on to other colleagues at the university. And this, a nice, boring summary of everything that was found. And all I can say is just, if you haven't done a BioBlitz before for your museum or for your institution, just do it. They are fantastic fun. Over the years, we went from about 50 to 60 experts and participants, surveyors in the first year, to I think twice that number now, over 120 people involved. And the public always take on, always come along in good numbers, as long as the weather holds. And just great fun all around. I want to thank iDigBio for funding, and thanks Spinach and Tedwig and our local organisers for hosting us all. And finally, let me get this right. Kiti kureti arigato. Thank you for listening. Any questions? Nope. I'll just wait a little bit so we don't jump the gun too much, and we've got 1 minute, but otherwise we've got, I
feel like I should ask a question. Two kind of related questions. Are you intending to go back and go to one in person? And have you started doing it? I mean, I imagine they're already in existence in Glasgow, but in Edinburgh, but have you
I've started a new one.
Yes.
I would love to go back to Trinidad. It's still on the list. I intended to go back much sooner, but then COVID and everything. I haven't started one directly in Glasgow. Because we've got other things going on in the university. We've got an online, we've got a iNaturalist project for the university campus, and I get involved in City Nature Challenges each year, and we run that. But yes, I'm ashamed to say I haven't done one properly myself in Scotland since I got back. Still sort of figuring that one out. Yep, I think we go.
Gavin Broad asks, were there any significant taxonomic gaps you would have loved to have filled?
Oh, yes. Many. The insects were always one that was disappointing. We did well on butterflies, well on moths. There's some good moth trappers. But the coleoptera were never done as much as I would like. The hymenoptera, the wasp, the parasitic wasps, the hemiptera. So lots of insects orders. We certainly needed more local experts on. The vertebrates are always very well covered, and the plants were fantastic as well. And it'd normally end up a battle between the vertebrate groups, the plant group, and everything else. It's 3 different groups to see who'd get the most, and they'd normally come fairly close to each other. But yes, certainly lots of gaps. Okay, right. I think if we can move on to the second talk, which is an online recording from University of Tokyo. Here we go. Yeah.
Hi everyone. Good afternoon. Thank you for joining us today. My name is Yurika Saito, and I am in charge of the Curator course at the Tokyo University of Agriculture and Technology. Today, I will talk about our BioBlitz lesson using iNaturalist, its educational impact, and the challenge we faced. First, let me provide some background. In museums, the distillation of biological data in Curator is essential for research and preservation. In Japan, a lot of data has been collected and is used for research due to the standardization by Darwin and JBEF. So, at our university, many students have a deep interest in wildlife. However, opportunities of acquire skill in handling biological data and understanding how to utilize it are limited. This project provided the opportunities to enhance, so I will present you the achievement from this project. We introduced the BioBlitz exercise, where students use their own mobile device to participate in the hands-on field activity. We started this in 2019, and it has been conducted every year with a variation in location and method. We have conducted this exercise in 2 different locations. One is a conservation area in the urban city, as a facility affiliated with the National Museum of Nature and Science. The other is our campus area in the local city. So here how the lesson works. First, we start with a lecture on digital data and how to use the mobile app. Then, students from teams and explore the field to experience a BioBlitz. After that, they gather to present the species they found. The group with the highest score wins a prize, Finally, we discussed their observations and ideas for the museum project. Now, let's take a look at how students conducted their species exploration. This is from the 2021 BioBlitz at Koganei campus. The students devised strategies and explored the campus. This photo is the students exploring around the campus. Of course, some students are not familiar with wildlife. This is bonus plants, Newton's apple tree. It is in an advantageous location for physics students. So capture the plants that they found on their mobile, so focus and shoot on. And so sometimes reptiles and so small insects. And after the BioBlitz data was integrated in each group, so counting the species they found. So finally, group 2 is one, and which found the bonus and was upset. This is other side date. Here the result for 1 year. The most commonly found species were plants, brought by insects and birds. According to student feedback, the plants were the easiest to find, but they found reptiles and insects and birds are the most exciting search for, even though their species were more difficult to find and photograph. So many students expressed that they enjoyed activity a lot, particularly the game-like aspect of species observation. However, some raised concern about the security of using social media, especially regarding the personal identification. Those location data and the need for guidelines of handling rare plants and rare species. Additionally, the students suggest involving children and the elderly in future events, as well as increasing opportunity to work with scientific names. This lesson was conducted in a very compact format, just 3 hours long. Despite the time constraint, the BioBlitz exercise proved to be an effective tool for creating a new educational curriculum centered on the use of biological data. Moving forward, it will be important to develop lessons focused on analyzing and utilizing the data will be collected. So if you have any questions or comments, please feel free to reach out us the contact information below. I would like to sincerely thank the students and everyone at the Nature and Science Museum, University of Agriculture and Technology, for their cooperation in this project. Thank you for all your attention. Thank you so much.
Arigato, Yurika-san. Is she online? Okay. Do we have any questions?
Thanks for Saito-san. Daisuke Sakuma. It's just a simple question. How do you think the identity accuracy for the Japanese system in your feeling by iNaturalist? Is she online now?
No.
Okay. Okay, I'll ask about it on the email.
Thank you. Okay. Well, the next talk is, I think, going to be Gavin Broad from the UK. Is he on? I think he's online because he asked a question earlier. But it's got a pre-recorded talk. So, yeah, when you're ready to go.
Morning, it's my pleasure to talk to you about DNA Bioblitzes, in particular how Natural England have teamed up with the Natural History Museum to collect invertebrates for genome sequencing and for DNA barcoding. So Debbie Leatherland is at Natural England and she's on the DNA monitoring team. Inez Januchak works for Darwin Tree of Life, based at the Natural History Museum. Ben Price is a curator of freshwater insects and working on DNA barcoding at the Natural History Museum as well. And I'm a WASP curator, but also heavily involved in Darwin Tree of Life, which I'll mention, and using those genomes downstream. But why are we collecting for Darwin Tree of Life? And what is Darwin Tree of Life? This is an ambitious project involving all these lovely partners here who are combining expertise to collect eukaryotic species in the UK and go from a specimen in the field to a complete chromosomal genome within a fairly short period of time. And this is for a huge variety of different species, from fungi to lichens to plants to animals. And at the moment, we're coming to the end of a pilot phase funded by the Wellcome Trust. So that's 5 years of collecting. And we have about 2,000 genomes assembled at the moment. And out of a sort of total of about 20,000 that we think we can realistically collect in the next decade. So we're doing well, we're on track. But that's not the only reason we're collecting for DNA. We have quite a pressing need in the UK for a reliable UK DNA barcode reference library. Although about half of the UK eukaryotic species are represented by some sort of DNA barcode, we only have about 4% of UK species represented in bold by a UK barcode. So that's clearly not good enough for the various applications of DNA barcoding for example, measuring the health of ecosystems, aquatic monitoring, that sort of thing. And although we don't have we don't have steady funding in place, there are various sources of funds which are creating this reference library piecemeal at the moment. So we're working with Natural England closely, and Natural England are involved in managing many of the national nature reserves in England. So that's about 221 of them spread across the entire country, representing about 0.8% of the land surface of England. That's 110,000 hectares, amazing network of special habitats. And these national nature reserves were set up in the 1950s to protect habitats, but also very explicitly to provide outdoor laboratories for research. And it's in that spirit that we are working with Natural England to collect species for DNA in these national nature reserves, and including parts of the NNRs that most people don't get to see. But why a DNA BioBlitz? Why do we need to sort of team up to do this? To collect a sort of wide range of really reliably identified invertebrates, often not particularly easy to identify, you need a good good cohort of collaborators. So we bring together groups such as the Natural England Field Unit, curators at the Natural History Museum here, and some specialist groups who can identify, who can, and of course the reserve managers are key to all this, the people who know these areas of land and can tell us where to go and where to collect. And we sometimes bring in some fantastic local groups as well, which I'll come on to in a second. How do they work? So like many BioBlitzes, there's a lot of intense field work, but unlike many BioBlitzes, there's also quite an for somebody like me, about half my time is spent actually at the microscope in in an impromptu study space such as this. This is actually a school education room at the Wire Forest Study Centre, which we turned into our little mobile lab with microscopes and -80 freezer. And we spend a lot of time trying to make sure that we're carefully identifying things live, because then they're going to get flash frozen at -80 or on dry ice, and then they're destined for genome sequencing through Darwin Tree of Life. Things that can't be identified live are preserved in ethanol and DNA barcoded. At the end of this, we have specimens, and we have a species list that we give to the reserve managers, which hopefully adding quite a few new species to those reserve lists. And sometimes we have somebody like the Wire Forest Study Group or the Liverpool Museum coming along, and their experts help us enormously. These DNA Bioblitzes started in 2019. It's a pilot project before we got the funding for Darwin Tree of Life. This is us going up to Ainsdale on the amazing sand dune coastal system in the northwest of England, close to where I grew up. There's a rare photo of me in the field with a net. So it was really successful. We wanted to build on the success of this. Unlike some subsequent BioBlitzes, we did some marine intertidal sampling, and we also set up a lab in an office there where we did DNA barcoding on site using a MinION, and that was fantastic for public outreach. We had a slight pause then as there was a pandemic and various lockdown restrictions, but eventually we got going again in 2022 when we were again in the north of England. We went to Hatfield and Thorn Moors, which are particularly precious for their bogs, peat bogs, which have not been much disturbed. Some really special species there. Then we went down to Bure Marshes in Norfolk, where we sampled fenland, a very different range of species. And then this year we were in the Wire Forest, mostly ancient woodland, but a great mosaic of different broad-leaved habitats. When we were joined in the field by the Wire Forest Study Group, who have decades of experience in recording the fauna and flora of the Wire Forest. It's great to be out with them. One of the results, on each BioBlitz, we generally catch around 400 specimens, and that represents about 300 to 350 species, which are flash frozen. We've got some problems with the data from Ainsdale, but we did collect more than 55 specimens, I can assure you. And many of these species are new for Darwin Tree of Life in each trip. We have collected some of those iconic species that we have targeted, like the northern dune tiger beetle, pretty much only known from that sand dune system in the UK, or the Maya pill beetle, Curomopsis nigrita, known from very few places in the world and only 2 sites in the UK. I love this field photo of using a phone down the microscope, which is why it's not great quality, but these are the exciting moments when you get what you're after, when you get a really poor quality photo. And we collect, this is a graph showing the orders of insects, invertebrates, that we collect in each of these BioBlitzes. As you'll see, there's a great bias towards Lepidoptera because moth trapping is fairly easy, and we usually have a few Lepidoptera experts. Hymenoptera, you can see I'm obviously getting better with each trip at identifying things, so that's nice. We've had a slight problem in getting dipterists along the trips, so we do need to concentrate on a bit more, but you can see there's a lovely spread of expertise and we're doing really well, but also areas where we can improve and get better coverage. But each of these trips are about as successful each time, which is really pleasing. It's good to know that that each habitat is of great quality. One of the key outputs, of course, is genomes. This is a genome note of for a stone-bited beetle that was collected at Bure Marshes in Norfolk. So obviously we published this genome with in a little paper which has all sorts of fancy graphs and statistics about the quality of that genome, and that's what we want. But another important thing about these BioBlitzes is getting together, sharing expertise, and forging links across different organisations. These are the teams in Norfolk. It really was a glorious collecting trip. With iconic species for the UK such as swallowtail butterflies flying around us, and we do find a tiny bit of time for socialising. And this is the end of a BioBlitz. So there you go. A rather perhaps not the most exciting event, but there is a dry shipper at liquid nitrogen temperature, lots of tubes being placed into that. The tubes end up in our -80 freezer back at the museum, and we get racks of legs. So we don't end up, obviously I'm a curator, but I now acknowledge that I'm curating a slightly different sort of collection. But these collections are growing, and the genomic resources are increasing all the time.
Okay, well, thank you.
Just to thank all the participants, and especially the reserve managers who've been amazing at putting up with us and indeed encouraging us onto the land they manage. And obviously our funders, do check out Darwin Tree of Life and Biodiversity Genomics Europe. Thank you very much.
Yeah, thank you very much, Gavin. That was fascinating, very focused, different type of BioBlitz. Are there any, I'm just going to jump in with a question, but are the plans to sort of go into Scotland or Wales or Northern Ireland, or is this very much an English project for the moment?
No, that's a really good question. Thanks. We have actually gone several collecting trips to Scotland. I just wanted to really focus on the collaboration here with Natural England, but we've collaborated very closely with Nature Scot, and we've been going fairly regularly to an amazing sort of national nature reserve in Scotland called Bonnahme. So yeah, Wales, we've got to do a little bit better at finding our partners there, but yeah, it's really important in a project like Darwin Tree of Life to get great geographic coverage. So a little bit of work to do, but yeah, sorry for focusing on England there.
No, that was wonderful talk. Any other questions from the audience or online? We have two people.
Hi, this is Takano from Hyogo, Japan. To whom should I don't know to whom should I address to, but may I ask the definition of BioBlitz, since I heard that was today for the first time, so I'm started confusing.
Right. So the definition of BioBlitz that I've sort of used over the years generally comes from the first event that was run in 1997, I think, in the US, and it's really just a focused, intense period of biological surveying over normally a defined time with a kind of public aspect to it as well. There's various definitions, there's various lengths. It can be any length, it can be any size, it can be cover any geographical area now, but it's really just a focused period of study of biodiversity. But there's obviously people that interpret it in different ways. People spell it in different ways. Some people capitalize both the B's, some people don't. So it's developed very much over the last almost 30 years now. So, but there's a very good summary on Wikipedia of the origins of the BioBlitz and where they've happened in the world. But they're ubiquitous now, everywhere. Another question for you, Gavin?
Right.
Hi, it didn't seem like you had public participation in this one, but I'm just wondering how you deal with permitting across all that area and all those different collaborators. Are you guys on like a blanket permit? Because one of the issues is getting public on permits to do that kind of DNA BioBlitz, which would be great, but hard in a lot of places.
Yeah, no, that's a really good point, and that's why they're not particularly public. The first BioBlitz, we did invite quite a variety of partners, quite a variety of people to come along from local universities and natural history groups, but it is difficult then it does create a bit of a added complexity with all the permits, material transfer agreements, things like that. So at the moment, we've tended to focus on just a few partners, and then we work out an agreement with Natural England and the landowners, if they're not Natural England, so that we get so we record acquisitions into the museum collections from each of those collectors. So yeah, it is a little bit tedious, but having Natural England on board, who manage a lot of the who are basically in charge of a lot of the licensing around collecting and use of specimens, that's really amazing for us, and it makes it a lot easier.
Yeah, and just to add to that, in Trinidad and Tobago, there wasn't the same issues. We got a permit from Wildlife Department for any specimens that were collected, but ultimately, when it came down to running the events itself, it just depended on where they were taking place, but people were able to travel all over the country and survey in different places. Some of them were government-owned, and we just have permission. We didn't have to actually have a permit, just permission from the government to go. Other ones, like the Acerite Nature Centre, it's just their land, and we could survey anywhere there. So not the same restrictions I know many other people might face in other parts of the world.
It's possibly also worth pointing out in Britain, at least, that I guess we don't have many restrictions on what you can actually do as long as you've got landowner permission, and we're not collecting heavily protected species.
Yeah, it's a little bit different in Scotland, where you have the right of access to roam across pretty much all of the country. Whereas England, yes, it's sort of looking for landowners' permission to access certain parts. Okay, well, thank you very much for that, Gavin. Do we have any more questions? No?
One from the previous speaker. It was the previous speaker's online and responding to the question.
Yep. Okay, well, thank you for, I think, getting up early. It's not too early for you, is it? So, good. Thanks for joining us.
Thank you.
We've got time for a question for the previous speaker. Yeah. A response. Okay, thanks.
Say that to the group here, thanks. Whoops. Okay, thank you, Professor Sakuma. The Japanese system was a bottleneck, but after a few years, it has become much easier to use. I hope you can improve the rewards and game elements after finding a creature. The train activity in the Japanese app was also very interesting. The timing of the coronavirus was very unfortunate. I think BioBlitz is better suited to searching for living creatures in urban areas of Japan than deep in the mountains. Then she says, sorry if I didn't hear your question properly.
Okay, thanks, Libby. Okay, I think we can move on to our fourth speaker. This is Viktor. Who's here?
Hello, everyone. I'm Viktor. I'm from Münster in Germany. And don't worry if you've never heard about Münster. I will briefly introduce you to the area. As you probably can recognize from the title, I might not quite fit into the section called BioBlitz and Museums Natural Partners, although both words, BioBlitz and museum, do happen in my talk. Museum does happen a lot, but I actually has a slightly different scope. So, but judge for yourself. I think it's fitting enough, but I had some troubles finding a section that would like really fit the talk. So finally I landed here. So I hope you don't lose time with me. Yeah, Münster. It's this red dot on the map that you can see. It's close to the Netherlands border. It's actually quite a big city. And the Natural History Museum locally, it's almost 150 years old. Our collection dates a little further back, like the oldest specimens we have are from 1830s. And it has always been focused on mostly on regional fauna and flora. We do, of course, have some exotic stuff as well, but regional focus is one of the one of the important traits of the museum. And we have around 100 people on the staff, but here everyone is counted, like student assistants, cleaning staff, everyone. So don't think we're like really huge. We're actually a medium museum. We're not a small one, we're also not a big one. And this is important. As you can also see that from our collection data, we have around 1.5 million insects and maybe several 100,000 plants, and also a couple of 100,000 mollusks and geological and paleontological objects. So we are not as big as Berlin or Munich or Hamburg, like large German collections, but we're also not as small as many others, because in Germany there is a plenty of small museums. So we are a medium one, which is why we can kind of understand what small museum predicaments are, but at the same time we're maybe big enough to implement some solutions. Because what small museums are often confronted with, in Germany it's not a rare case when you have a house with like 5 or 7 people on the staff, the director, somebody for science, somebody for pedagogics and knowledge transfer, maybe a librarian, maybe a couple of other people, and that's it. And this scientific staff has to do everything. conduct research, doing knowledge transfer, managing the collection, like everything. And in this case, of course, people start asking themselves, how do I do this? Can I involve citizens? Can I involve volunteers? Of course you can, but nowadays people, of course, start getting deeper with this question and asking themselves, can I involve citizen science? Can I involve online citizens? And this is where we come in. We tried to use one of these online biodiversity platforms for like reorganizing or integrating the whole fields of the museum activities that I try to sketch here in brief. And the platform that we've been using is observation.org. It's like, I heard a lot about iNaturalist here on the conference, but nobody yet mentioned. I think once. I think in one talk it did happen, but observation.org seems to be less known in this part of the world, which is not surprising because it's originating from Netherlands. So it's European based, and it has been restricted to Europe for quite a while, but now it's operating worldwide. And it actually has more data than iNaturalist does. 255 million data set, which is quite impressive. I think iNaturalist has something around 200 at the moment. And they do have AI pre-identification of things, like you upload a photo and AI tells you immediately what kind of species it is, and it's getting better and better. It's now performing like really good. And the one trait that we like most about this page is that they have this human validator stuff. So you only get an like research quality mark on the data if you have a human validator like confirming that this species ID is correct, which is differently handled from iNaturalist. So this is the feature that we like most, and we have been doing a lot of work with observation. Like, for instance, research has been, it has opened some new possibilities to us. I just show you a couple of data. I'm a Heteroptera person, I work on Heteropterans, and I presented a poster on this data that we, in ICE in Kyoto a week ago. So what we did here, we conducted a Heteroptera challenge throughout Germany, and it was quite an amazing success. For instance, if you can see here, this is the Heteroptera records before the challenge in the city of Münster, and these are the records after the challenge. So you can see the boost is quite obvious. Here you can see some geographical bias, so the most users tend to concentrate, the closer to the Netherlands border, the more users, because like it's been filtered through the border to Germany. But it's an amazing data set. The data set on Heteroptera on this portal has been tripled during the challenge, just in 8 months. And we had a lot of data to play with. Like, for instance, I've been comparing this data. This is just the week, days of the week and activities of the people during the challenge. And you can see here, people have been most active during the weekends, like on Saturday and Sunday. And funny thing, I compared our results with a paper on similar activity in Spain, and the people were most active there during weekdays. So, like, Germans would upload the photos on their weekends in their free time, and Spaniards obviously on the weekdays. Well, people are different, and sometimes not implying anything, Actually, also here, we can see that the higher the temperature, the more are people prone to uploading data, which is probably also would be different for Spain, because I don't think when it's 40 degrees Celsius outside, people would go much making photos. So it's pretty interesting to compare with different stuff. And this is just a brief comparison of different habitat affinities of the species that were registered during the challenge and before the challenge. But this goes too far. If you have questions on that, I'm glad to discuss that with you later. But anyway, we've been using it a lot. We start using it a lot in the research. And another activity that Natural History Museum does is, of course, publishing the results. And there are two ways of publishing like occurrence data. For instance, citizen science platforms, just like Observation or iNaturalist, they're perfect, they're very up-to-date. As soon as you see something, you publish it immediately, and this is the most up-to-date data that we have. But they're sometimes not that reliable, because AI can misidentify stuff, people can misidentify stuff. So that's a little bit of a problem there. And scientific publications, otherwise, they can be very careful, but they're not so up to date. They are like always they're hanging backwards like maybe five years or something. So we are kind of starting to think, what can we do to bridge these two different pools of authors and pools of readers and users, because they're of course they cut each other. There is some users that use both sources, but not everyone uses both. Sometimes people just concentrate on online platforms. Sometimes people just read paper written stuff and nothing else. So we try to implement in our two publication organs and our two journals, two on biology, we also have one on geology and paleontology. We try to start a format, or at least starting to regularly publish stuff on the results of the that we get through observation.org. And this is just one publication, just an example of the data on the praying mantis religiosa, spreading in Germany like 2012, 2020, 21, 22, and 23. And yeah, this is just an experiment, and it's, well, we still need to work on that. Yeah, but at least we think this is a very, very interesting idea for the future. And yeah, what I also try to do with observation.org, I'm trying to use it as a digitization platform, because we do not have that much stuff. We are a medium-sized museum, but I would like to have like maybe 5 or 6 people. I only have 2 students to work with, so it's not that we can move mountains with that, but they do a great job, but we're just too few. So we don't have any solutions employed already, any databases or something. So I was thinking of this platform as a sort of a data bank that is already there. You have a structure, you can upload the photos, you can upload the metadata, and we just thought, why not try it? So we tried that. We're conducting actually at the moment a pilot study with Apollos and Caribbees, Apollo and Caribbees just as an example here. And well, why would I do this? First, one thing is, as I said, there is an infrastructure already. And the second one is, you probably recognize this graph here. This is from a seminal study by Hartung et al., which like gave a big push to the discussions of insect decline. And this study was published in 2017. At least in Germany, it has been really a seminal study, because until then, people were not talking about that publicly. And after that, it just spawned a great amount of studies and governmental programs and stuff. And one of the reasons why it's also a seminal study is that they were relying on volunteer data. They were not employing professionals. They were just so-called amateurs doing the job here. And the problem is, you see, the data starts around 1990. So what happened before, we don't really know much about that. And this is the problem we have with natural science platforms like iNaturalist. We only have like last 20 years of data to compare with, and what happened before we don't know. So I thought, when we can convert the scientific data from the museum collections, from the specimens, to these portals, then we can compare them like simultaneously. So this is why we're doing it, not only because there is an infrastructure ready, but also because we can compare the data really good. But of course, there are some problems with that, like assignment of localities is a little bit complicated. For instance, in a collection, if you have a specimen, you are very lucky if you have a specimen with coordinates. Very often you have a specimen that just says Münster, and sometimes it only says Germany. So have fun with that. There are solutions you can apply here, but we're still working on that. And there are some other problems. I think I'm running out of time, so I will not detail them here, but you can ask me afterwards. Yeah, and one last aspect where the word BioBlitz finally happens is, of course, we can try knowledge transfer with observation.org. And we are starting a pilot project on a school nearby. There is a gymnasium, sort of a high school, with 8th grade students. And we kind of, preliminary called it Schulblitz, school blitz. This is a competition among schools. So we will ask several schools the simple question: whose backyard, whose schoolyard is most species? Who has the most biodiversity there? So we'll just send the people out there to make photos, and in the end we'll see who will win that. Yeah, the picture actually does not show school children, but these are real people. It looks a little bit like AI created, but it's not. Yeah, it's. Yeah, so it fits very well with school curriculum because it's the 8th graders when they start doing the ecosystem connections, and they also kind of acquire the first digital media competence, so this fits very nice. So the teachers are quite happy, and after I come back from Japan, we meet in October for the first project meeting, like with all participants, and we start a pilot in autumn, and the Blitz itself starts after Easter 2025. Probably not a sentence a German person should say out loud, but yeah, the project will start 2025. And yeah, conclusions. I think that such biodiversity portals like observation.org have really nice potential for reorganizing and also integrating the activities of a natural history museum. And for research and for education purposes, they're quite ready to use, in my mind. As for publication and collection management, of course, I've tried to highlight a couple of problems that I see there, and there are some, but I think there is still potential. And what is important, I think one important thing is to this integrating aspect, because sometimes the problem with the museums is that many activities sometimes run like differently, and people do not know about each other, what they're actually doing, and this could help us to, to move things together. Yeah. So I would like to thank a couple of my colleagues that helped me with scientific stuff, and are people who work in the collection, also the photographs that were provided by several colleagues. And I would like to thank you very much for your attention.
Okay, thank you very much, Victor. That was very interesting to hear about other observation platforms. I'll certainly be having a look at observation.org when I get the chance. Do we have any questions from the audience or from online? No? Okay, I think I'll just a couple of things. One was you're saying the observation.org, the research grade, shall we call it, classification. You're saying that it takes a human to actually verify what something is. I mean, that is the same on iNaturalist. You need two people to verify. You could be using AI to give you a clue, but then you need someone to sort of still agree with it. Yeah. But there is the risk, I guess, of someone else just using the AI to identify as well. But how does it work on observation.org for clarification? So on iNaturalist, you have two people, just anyone on the web can just, if two people are agree on something, then it's considered research quality. In observation, there is a human validator that has been appointed offline. So there is, this is an expert on some group that has been appointed by organization offline, and this person or persons, it's ideally you have a whole group of persons, this one of them or many of them has to confirm that this species ID is correct. So it's, well, I know cases from iNaturalist where there are two people who are not specialists on the group, and they say this is definitely the species so-and-so, and then a specialist who is alone comes and cannot do anything because two people are agreed on. Of course, there are cases where you can correct this, but this is sometimes a problem. Yeah, no, that is definitely one of the big issues with iNaturalist. And in the UK, one of the major platforms used there is iRecord, which evolved out of the various biological record centres around the country. And they have that same verification system where an expert, recognised expert, has to confirm a record before it then gets shared with bigger platforms like GBIF and gets accepted. So yeah, that extra level of verification is there. I think one of the main reasons I used iNaturalist, even though I'm UK based now, is that there wasn't any other option when I was first started doing this in Trinidad and Tobago, and same with many other countries around the world. It's a very good global platform, despite its flaws at the moment.
Yeah.
But yeah, certainly it's more work required. But I think with any of these things, when you want to analyze the data and do something with the data, it's still reliant on you as the scientist to investigate the data and decide what is reliable and what is not. You can't just take it at first glance and assume it's all accurate. But okay, do we have any more questions? No.
It's not really a question, just a comment. You mentioned BioBlitz as in Wikipedia. As far as I can tell, it's not in Japanese. So I looked at where it's defined, and there's clearly a whole bunch of languages where it's not defined. So the notion that it's a new term is probably quite, obviously it is for some people in the audience. So maybe one sort of mini BioBlitz activity would be to make sure that different language Wikipedias have a page to define what a BioBlitz is.
Okay, yeah, that sounds like, I'm just going from what I read several years ago now, when I was making up my own university BioBlitz page, and I took the sort of one-line definition from the Wikipedia page to use on the university one, and I must admit I haven't looked back at it since, so, but yes, a global definition could be useful. Okay, well, thank you very much, Victor.
Thank you.
And we now have our next speaker, please.
All right, thanks everyone for sticking around to the end of the day. My name is Lauren Vonnahme, and I'm a senior museum specialist at the American Museum of Natural History in the Department of Herpetology, and I'm here to talk about the role that BioBlitzes can play in museum community partnerships. So, different from a traditional biological survey, a BioBlitz is a community-driven citizen science effort to record as many species within a designated location and time period as possible. Glad we finally have a definition. They're often used to get the baseline biodiversity information or record biodiversity changes over time. Essentially, public participation is what makes the difference in a BioBlitz versus traditional fieldwork. Natural history museums are ideal facilitators for BioBlitzes, as they have direct access to expertise and equipment unavailable to most communities, and they employ teams of scientists and educators that can directly link biodiversity inventories to the scientific processes and specimen collections. Museum-sponsored BioBlitzes can also be a popular tool for breaking down barriers to science and helping create equitable access to scientific processes. And today I'll be presenting a BioBlitz used by scientists from the American Museum of Natural History to help create experience for students from underserved communities to get valuable access to nature. Our BioBlitz quickly becomes one of my favorite events to work with every year, and I'm really hoping that we can engage other museums to promote this kind of thing. And all the photos you'll see will be from our students at our BioBlitz. So as many of us already know, there's a lack of diversity in the STEM fields, particularly getting young students introduced or interested in the sciences. For instance, students from lower socioeconomic backgrounds often face barriers in developing a strong STEM identity due to systemic biases and stereotypes. Interventions that focus on inclusivity are essential, and programs like BioBlitzes and natural history museums can provide equitable access to STEM experiences for young people from marginalized communities. By engaging these youth in scientific activities, such initiatives can help them envision themselves as future scientists and contribute to a more diverse and inclusive scientific community. Natural history museums like the American Museum of Natural History are determined to do what we can to shorten the gap and foster an environment of scientific curiosity across several demographics. So, located in New York City, the American Museum of Natural History is one of the world's largest and most visited museums, with around 5 million visitors annually. Behind the scenes, the museum houses over 35 million artifacts and specimens across multiple divisions of vertebrate and invertebrate zoology, paleontology, anthropology, earth and planetary science, astronomy, etc. Along with a full-time staff of around 1,000 people, including scientific staff like curators, postdocs, research staff. We have around 1,400 visiting researchers a year and loan out around 10,000 specimens. So the AMNH is a huge resource for the scientific community. The New York City metropolitan area is one of the most diverse cities in the world, with over 20 million people and over 800 languages spoken as the primary language. That number blows my mind. Unfortunately, the diversity of the museum, particularly in the sciences, does not match the diversity of the city, which is a well-known problem in the STEM fields. So in 2020, AMNH launched a 5-year strategic plan in which we set long-term commitments for DEI initiatives. These include things like building and sustaining a diverse and inclusive community, addressing barriers to the full inclusion of historically underrepresented groups, and ensuring an environment where all members of the community are engaged in our mission. In line with the 5-year plan, the AMNH has developed programming focused on addressing the critical need to build diversity in the sciences through better outreach and recruitment for our PhD and undergraduate programs. We have professional development for New York City school teachers in order to foster diversity and equity in the classroom. We expand efforts to engage diverse participants in internships and workforce programs at the museum. And we also sponsor New York City school groups and other public programming that may not be able to afford the museum free entrance to make sure that the museum's commitment to the community is more inclusive. So inspired by this, curators from the AMNH, curators and scientists, began our own BioBlitz to support students from underserved communities to get valuable access to nature. So why reinvent the wheel? To ensure the effectiveness of such a program, the curators from the AMNH have been working in partnership with the Renaissance Youth Center. The RYC is a nonprofit community organization that focuses on empowering youth through a variety of educational, artistic, and civic engagement programs, with a heavy focus on STEAM initiatives. And their work in the community is critical. The RYC is located in New York's 15th Congressional District. That is the poorest congressional district in the United States. Only 22% of people in this district have achieved at least a bachelor's degree. That is significantly lower than the state average of 40%, and only 79% of students graduate high school in this district. And 99% of the students at the RYC are considered Title I, which means they come from a lower socioeconomic background. And this program serves over 4,000 students weekly to provide a safe and supportive environment for personal and academic growth. It's a really cool program. They do things like tutoring. They have classes in agriculture, zoology, architecture, astronomy. They do coding for robotics and computer programming. And their program significantly impacts high school graduation rates. So students who remain there for 3 years are more inclined to pursue sciences in their future. And students who remain for 5 years have a 100% graduation rate, which is insane. This is an amazing community program, and it's actively changing lives and bringing science to students who would not typically be introduced to STEM programming at this level, and they are the perfect partner for scientists from the AMNH. So how do we bring our scientists' expertise, the AMNH's vision, and RYC's STEM mission together? Allow me to introduce the RYC BioBlitz. So our BioBlitz started in 2022 after one of our curators was visiting with friends upstate and realized that their large mountain property would be perfect for a BioBlitz. And the owner was like, yeah, I'm all in. What's a BioBlitz? So as a longtime supporter of the Renaissance Youth Center, they started devising the plan to host a BioBlitz for the RYC students. So for the past 3 years, we all meet about a 2-hour drive in upstate New York in the Catskill Mountains for a weekend of camping, nature, and scientific discovery. This is an incredible location. It hosts a wide variety of East Coast flora and fauna, and introduces kids to nature in a way that is not achievable in the urban neighborhoods of New York City. I don't think people realize that New York has mountains, but this is an incredible natural mountain preserve. It's truly a life-changing experience for these students, and it's a highly competitive selection process. So out of 4,000 students, only 10 to 12 get chosen through a merit-based system. Students are not allowed to skip any classes. They can't miss their after-school programming, good grades, no bad behavior. They even compete in an annual science fair in order to try to get a spot in this. One of our students from this year, she wanted to come so badly, she worked the entire year on the science fair project. Don't worry, she got in. So prior to our BioBlitz, not one of the selected students had been on a camping trip before. Most of them have never even left the Bronx. So we outfit the students with camping supplies, field gear, professional ID guides, species checklists. While they're with us, they learn how to properly catch, handle, and ID animals. They learn the scientific language so they can leave confident in their ability to speak about specimens and field work. They use their guidebooks to help them identify the animals and their specimen checklists to track and inventory the number of species they catch over the weekend. And under the direction of the curators, the students learn about the different types of projects that scientists can work on in the field. So here are some of the examples of the amazing hands-on experience they get. So with Dr. Stiasny, who's from the Department of Ichthyology, and with her postdocs, they learn how to seine net for fish in the Esopus River. We got some pretty cool stuff this year. With Dr. Jessica Ware and her team, with Dr. Smith, who's the curator of ornithology, they learn how to set up mist nets for birds, and they can identify the difference between males and females and adults and fledglings. And this is actually a big pathway for migratory birds, so the students really learn a lot and can catch some pretty unique species. With Dr. Jessica Ware and her team of postdocs and graduate students from the Division of Invertebrate Zoology, they learn a ton of field techniques for the hugely diverse group of inverts found. So they do things like dip netting for aquatic invertebrates, sweep netting for insects in the tall grasses, setting up light traps at night to look for some nocturnal things, and a fan favorite is to use the blacklight flashlights to look for the fluorescing centipedes at night. The kids love that. And with Dr. Birmrink and myself from the Department of Herpetology, we spend a lot of time flipping rocks and logs and looking for the dozens of species of frogs, snakes, and salamanders native to the area. I may be biased, but I think the snakes are usually the biggest hits of the weekend, but mostly because it's something that they're not in New York, and there's a big fear around snakes. So when the kids learn to catch them and handle them and can go back and show their parents that they caught, a 5-foot rat snake over the weekend, it's a big hit. And of course, there's team building exercises like the near impossible task of building a giant tent, playing outdoor games, making s'mores, and another big favorite is the night of scary stories around the campfire. Following the BioBlitz, the students are all invited to visit the AMNH, where they tour behind the scenes and learn about the significance of museum collections and how they are used in scientific research. And this is a great holistic way for the students to make connections between the fieldwork and the research. For example, many of the students in the RYC coding program, they had no idea that you need to code to do research. And it's really cool to see how their passions can become career paths. So they think coding is a hobby, they can make a career out of it. Prior to the RYC and the BioBlitz, the student selective have never been exposed to this level of science. So I really want to highlight some of the attendees' incredible achievements. 3 of the 4 youth counselors who have attended with the students are pursuing or have gotten their teaching certificates in science. Zoe here did a science project on her BioBlitz experience and ribboned at the District State Fair. Johan, who loves to code, was selected to speak at the United Nations Media Summit on the impact of coding and STEM and after-school programs. And Jonathan here, he submitted a project to the National STEM Foundation and was a finalist. 95% of the students who have attended have reported an increase in their math and science grades by at least a letter grade. And all of the participants have credited the RYC and the BioBlitz as their introduction to the reason they are now interested in the sciences. I want to close out with a deeper dive on the benefits of working with external partners. With this immersive scientific experience, RYC and the AMNH can advance their missions of providing educational opportunities that nurture a sense of scientific curiosity. This initiative highlights the importance of addressing the historical lack of diversity in the sciences by providing equitable access to natural history opportunities and empowering young people from marginalized communities to envision themselves as future scientists. And you don't need to start from the ground up to create this kind of program. It's outside a lot of our wheelhouses and incredibly daunting, but programs like the RYC exist. You just need to show up with your passion and expertise, and you can make this happen. You don't need to deal with the logistics. You just have to show up and be like, here's how you ID a snake. And community programs like the RYC already exist, so you just need to partner with them. And I encourage you to scan the QR code. That's a video that the students made from the 2023 BioBlitz, and you definitely get a lot of good communication from them, talking about it's important. One of my favorite things is one of the students, the reason he liked to go wasn't for catching snakes. He just said he felt good in nature, and he didn't know how to explain it, but just like being around trees and being around grass, it made him feel good inside, which is like kind of sad for, the inner city of New York. Anyway, if you have any questions, feel free to contact me. We obviously have a lot of acknowledgements to the crew from RYC. We have a huge crew of donors and sponsors. And yeah, thank you.
Okay, thank you very much, Lauren. That was wonderful. So many similar things as far as what gets people excited from my BioBlitz experiences, things like the black light, looking for scorpions in particular, always got the biggest group of people following them around. And yeah, snakes, always snakes. So do we have any questions in the room?
Great job, love that. So I'm curious, you talked about this very intense process for these kids to, having to apply, getting picked, yada, yada, and it's a small group. Has AMNH ever thought about just going in your backyard and doing something bigger and more accessible?
Totally.
Right at
So-
Central Park?
Yeah, so that was one of the points I wanted to make, is that this is obviously selected. Like, we're blessed with a donor who's like, please come to my property and work with this program. But you don't need to do that. We've been planning it. We are thinking about doing day trips instead of big camping trips where you can bring the bus of like 30 students. We do BioBlitzes in Central Park. Like, you can do this in their backyard. There's actually some really incredible places, even in New York City, where you can find a snake a 10-minute drive from where this area is located. So this is just a cool big one that we love to do, but you can do this smaller with more students. Day trips are much more accessible. With this, you have the issue of parents letting their kids go away for a weekend, and that's all on the RYC programming, but we can make this happen in a smaller time, smaller space, closer to home.
Okay, anyone else? Anyone online? No? Okay, I think we're good then to go for our final speaker, but thankfully, a final round of applause for Lauren, please. Thank you. And our final speaker is, she's unfortunately not able to join us online, but she has a pre-recorded talk for us now.
Hi everyone, my name is Lila Higgins. I am the co-founder and co-director of the City Nature Challenge. I'm also the co-senior manager for community science at the Natural History Museum of Los Angeles County. I'm here today to talk about the City Nature Challenge and what we've been able to accomplish in 9 years. And that's from the small and local to what we're doing in Los Angeles and all the individual organizers, what they're doing locally, to what has been accomplished globally in this really large project. I am here presenting on behalf of the rest of the global organizing team, which includes myself, Allison Young from California Academy of Sciences, who's the co-founder and co-director with me, Sam Tyag, Amy Jecker-Jones, and Olivia Van Dam. The Natural History Museum of LA County and the California Academy of Sciences organized the City Nature Challenge, and we've been doing that from the beginning. So how did it all start? It started back in 2016 as a competition between LA and San Francisco, which city can find the most nature. It came about because the Obama administration designated the first ever Citizen Science Day. And Allison and I talked to each other on the phone and said, what could we do that would be fun, interesting, and not too much work for ourselves? And we both used iNaturalist, and so we decided a little competition between LA and San Francisco, which city could find the most nature, using this iNaturalist platform would be a good thing to do. We had a phone interview with a local reporter from the LA Times, and she asked me, Lila, how many observations do you think you're going to get? And I blurted out 10,000. Sounded like a good, big, round number, quite aspirational. And then Allison awkwardly laughed, well, we want 10,001. And lo and behold, we were able to meet that goal very nearly. We got 20,000 observations all told after the results were announced. But the results were announced, and we got 19,800. We had 1,000 people who took part, and 2,544 species were found. So many other cities were paying attention in the US and thought it was really interesting. And they said, hey, we want to do that. Are you going to do it again next year? And so we went national in 2017. 16 cities across the United States, more than 4,000 people taking part. We made over 125,000 observations. And then, of course, what do you do after you go national? You go global. And again, this is all word of mouth, organic growth. We have not paid for any advertisements around to get new cities to sign up. It has all been organic. In 2018, we had 58 cities around the world take part. We made 17,000 observations, 441,000 observations together with those 17,000 people. And it was quite amazing. 2023, we have gone up to the millions, 1.87 million observations, 66,000 people taking part, and we had over 400 cities. This last year, our 9th year, We made 2.4 million observations, 83,000 people coming together, 65,000 species were documented. And you can see just that vast number of cities in India. So I really want to say thank you to Vijay. He came on board in 2023, got about 20 cities in India on board. And then this year we worked with another local organizer, a local regional organizer, to get more cities in India on board. And we had over 200 this year. It's been so fantastic. I'd be remiss if I didn't mention Japan and the participation that Japan's had for the City Nature Challenge. Tokyo has been a long time organizer. Since 2018, they have been involved when we went global. And this year in 2024, we had Tokyo and Ishikawa, and they made almost 5,000 observations together, 219 observers. We're really excited to see more cities come on board in Japan. And of course, we've never had a city take part in Okinawa, but maybe this 2025 will be the year that that can happen. So what are the goals for the City Nature Challenge? The City Nature Challenge didn't have any goals explicitly when we first started. Again, we just want to do something that was fun. But after a few years, we're like, we need to write down these goals. And so we have 5 goals. We want to connect people to their local nature in their urban and metropolitan environments. We want to connect people to each other by building community in person and online around local nature. We want to help collect urban biodiversity data that's available to managers and scientists. It's open source on iNaturalist, so anyone can download that data. We want to grow volunteer biodiversity documentation globally through this community of community scientists, and we want to have fun doing it. We know that there are a lot of folks who are motivated by competition and a lot of folks who are more motivated by collaboration. We did an evaluation in 2018, and so we knew it was about really meeting people where they're at. And so we were able to meet people who are into the competitive aspect and people who are into the collaborative aspect in this one project. So what are some of the impacts of this 9 years of working on this project? So there's the numbers overall, growing from 2 cities to 690, from 20,000 observations almost in 2016 to 2.4 million observations. All told, it's over 7 million observations of urban biodiversity data that have been uploaded to iNaturalist. And about 100,000 people taking part globally over the years. You can see here is a graph from iNaturalist, the number of observations uploaded per week. And you can see it just keeps getting higher and higher. And the numbers never go back down. There is increased usership on iNaturalist because of the City Nature Challenge. Here's the active users on iNaturalist. And you can see there's a peak, maybe a small peak in 2016, but there's a peak every year. And it does dip back down, but it never goes back down to what it was. There's a new normal, and there's more increased active users on iNaturalist, collecting more data for us and scientists around the world to use. So City Nature Challenge could not happen without the people. We have about 2,000 organizers we work with globally. Many of them are pictured here with their participants. And so, yeah, it's these global organizers who know their community and know their cities best, and they engage with their local people. And they get them out to make observations, take them out moth lighting events, do observations with children. They do identification parties, sitting around their computers, maybe with pizza. So many different ways to engage and take part in the City Nature Challenge. And it's really powered by the people. We wanted to get to know a little bit about what the people are thinking and doing in the City Nature Challenge, and we did an evaluation in 2018, and we asked some questions about what were their motivations for participating, and about 36% of people said that it was because they wanted to have a learning experience, 22% said they really wanted to collaborate with like-minded people. Some people, it was really important because they felt like they wanted to help the environment and contribute to a cause. So there's lots of different motivators here. And then we also asked, as a result of participating in the City Nature Challenge, what did they feel like were some of the outcomes? 95% of people felt they had learned something new about the animals and plants specifically in their local area. 73% of people also felt like they had learned about organizations and projects that were working to monitor and protect animals and plants in their area. So that was like that piece of bringing people together and building community. So we knew we were on to the right track and meeting some of our goals, both with the quantitative and the qualitative results of these evaluations. Switching gears slightly, here's a few stories about some of the specific impacts. So in 2020, the City Nature Challenge was going on, and this white-spotted slimy salamander was documented in Arlington County around Washington, D.C., and it was found by a high school student. This salamander had not been found in that area since 1977. It was thought to be locally extirpated, but this high school student, Luca, was able to find it and document it and let people know it's still there so that conservation actions could happen. This is an observation of a orchid. It was found in Honduras, also in 2020 during the City Nature Challenge, and it was the first time it had been recorded on iNaturalist and on the Global Biodiversity Information Facility. So this was a range expansion for that species. Again, locally extirpated range expansions, these are the sorts of things that can be documented during the City Nature Challenge. Many papers have come out about the City Nature Challenge. These are all papers that are specifically about the participant engagement. And there's also been some papers coming out more alongside of how local governments can use this data and engage the public around biodiversity on a local governmental level. They did a really great infographic. I definitely check out this paper. They were looking at what the known biodiversity was before CNC, and then how much they could update biodiversity through CNC, finding things that maybe didn't know they were still there, refinding them, kind of like the slimy salamander, or finding new things that maybe are like introduced or invasive species. Biophilic Cities has also named the City Nature Challenge as a way for their partner cities to engage in eco-literacy and also through the collection of data. I'll just end us off with a little quote from one of the folks in Prague. It was such a fantastic way to learn more about the plants and animals that I see often, but I couldn't name and didn't know anything about. It was also great to connect with other people in my area who were participating in the City Nature Challenge. This just demonstrates how much it really is about people getting folks together and learning together about our urban biodiversity. Here in Los Angeles, we have a number of partners. So again, going from that global back to local, we have 30 or so partners who come together. We have all of our flyers in these 8 different languages. We really want to make sure folks get the chance to engage regardless of what language they speak. And over the years, we have had almost 10,000 observers taking part and have made over 193 observations. There are so many stories wrapped up in all of the years and all of the events and all of the partners that we've worked with. We don't have time to do that, but I really wanted to share that it really is a really small event for some cities and then this global event. So it really plays with that global and local. So all told, we have over 42 languages that the City Nature Challenge is translated into. We work on logos for each of the cities who need new language translations. There are also all these spin-off projects that have used this model of this global scaled BioBlitz. So for instance, there's the Hong Kong Inter-School City Nature Challenge that happened in 2020 and happened for a number of years, engaging high school students. And as you can see, they made a lot of observations. And then there's projects like the Great Southern BioBlitz. We have so many participants who really said, you know, this timing does not work well for us. We would love to take part in the City Nature Challenge if you had it in the fall. We obviously don't have the capacity to run 2 City Nature Challenge years. It is so much work. But it's great to see this spinoff from the Great Southern BioBlitz. And they are going to be doing another one this year at the end of November. And you can see all these observations, all these cities taking part in the Southern Hemisphere. This next year, 2025, is going to be our 10-year anniversary. We're going to be bringing the world together for biodiversity. We're going to make a big splash. It's going to be April 25th to the 28th. We're going out and making observations over that long weekend. Going to have time for folks to upload their observations and do identifications, and then the results will be announced on May 5th. We would love to have you be a new city. You can go to our website at citynaturechallenge.org or .com and sign up. There's a sign-up sheet there if you want to come on board. We are seeking global partners and sponsors for the 10-year anniversary. We want to do some really cool stuff this year. Myself and Allison are going to be going to the COP16 in Cali, Colombia, and representing the City Nature Challenge there. And we are working on more research using the City Nature Challenge data, and we would love for collaborators on that. So if you have any questions, again, my name's Lila Higgins. I use they, them pronouns, and you can email me at lhiggins@nhm.org.
Well, hey, I'll say thank you, Lila, even though she's not here, not online. Such a shame she isn't online, because I'm sure there would be many questions. I certainly have a few. We are pretty much... Yep.
There are some of us from Natural History Museum here. We may not know...
Sorry, on the microphone.
There are some people from Natural History Museum here. We may not know the answers to your questions, but we're happy to take them to Lila or see if we can answer.
I was going to email her anyway after the session just to let her know it all went well, and hopefully she will be able to watch the recording when we get back. So thank you all very much for sort of joining us this afternoon. And before we finish, I just want to give a little update on our current conference, BioBlitz, which has been doing, I think, pretty well. And very much thanks to Vijay Barf for setting up this event, as he's done many times for TEDWIG in the past. So at the moment, we have 2,319 observations made consisting of 671 species, 167 identifiers from around the world, and 58 observers from within participants. So I think that's pretty good. And seeing as how several people have already said they'll be uploading stuff when they get home, I wouldn't be surprised if we double that number quite easily before the end of the event. I think we're going till September 11th. Did you put it for? Yeah, September 11th for any observations, just to give people time to get those little extra trips in after the event finishes. But yes, wonderful. Well, thank you so much, everybody. And, well, beach party time soon, so. Cool.